Okay, so today's lesson we're going to focus on the light independent reactions. Just a quick review from last class. We saw that the light dependent reactions rely on water and light energy, and the end products were oxygen, and the intermediate products were ATP and NADPH, which will now be used in the light independent reactions to produce glucose. All right, so that is a quick summary of what we looked at last class. All right, so the light independent reactions of photosynthesis require that those products that we saw in the light dependent reactions. So we need that NADPH and we need that ATP that was produced. So when there is a sufficient amount of NADPH and ATP in the stroma of the chloroplast, the energy of these molecules will then be used to make glucose in the presence or absence of light. It's also called the Calvin-Benson cycle, the series of reactions by which carbohydrates are synthesized. So we're going to go through this model and we're going to go through it step by step. So step one is called carbon dioxide fixation. So step one is happening right here. So the carbon atom in carbon dioxide is chemically bonded to a pre-existing molecule in the stroma. It's this one here. It's called ribulose biphosphate. It's a 5-carbon compound and it's shortened to RUBP. So the resulting now compound is a 6-carbon. The resulting now is a 6-carbon compound, and it's very unstable. So this it's very unstable. It will immediately break down into 2 identical 3-carbon compounds. All right, so 2 of them. Now, because these 3-carbon compounds are stable, they're the first stable products of the process, the plants that demonstrate this process are called C3 plants. Summary of the reaction that leads to these 3-carbon compounds is here. So we have carbon dioxide plus the RUBP forms the unstable C6 compound, which forms 2 of those 3-carbon compounds, which are now stable. Step 2 is reduction, which is happening right here. This is now going to take that newly formed 3-carbon compounds. They're in a low energy state, and they're going to get converted to a higher energy state by, they first get activated by ATP, and then they get reduced by NADPH. And the result now of these is 2 molecules of PGAL. So right here, and PGAL is glyceraldehyde 3-phosphate. So now some of these PGA molecules are going to leave the cycle and be used to make glucose, and the remaining are going to be used to move on to the 3rd stage, which will be to remake RUBP. So step 3 is replacing RUBP. Most of the reduced PGA molecules are used to make more RUBP. So energy supplied by ATP is required to break and reform the chemical bonds to make the 5-carbon RUBP from the PGAL that we saw here. So this cycle has to be completed 6 times to synthesize 1 molecule of glucose. So of the 12 PGA molecules that are produced in 6 cycles, 10 of them will be used to regenerate RUBP, and then 2 of them are used to make glucose. All right, so we'll stop here and watch a couple of these videos, and then we'll do another quick summary of the Calvin cycle, or the light independent reactions. All right, so just a quick review then of this Calvin-Benson cycle. So light independent reactions, they don't directly depend on light energy in order to proceed. So they can occur in the daytime or during night. The products from the light-dependent reactions, the NADPH and the ATP, they move into the stroma of the chloroplast and then provide energy for the light-independent reactions, which is also known as the Calvin-Benson cycle. And then that energy in those 2 molecules is used to make glucose. So the 3 stages we talked about, first was carbon dioxide fixation. Carbon dioxide bonds to a larger carbon compound in the stroma, that RUBP. It forms a 6-carbon compound, which is very unstable. So it'll immediately break into 2 3-carbon molecules. The 3-carbon molecule then is activated by energy from ATP, and it's reduced by NADPH to form glyceride 3-phosphate, or the PGAL. Some of those PGAL molecules will then leave to form glucose, and the last step is that the remaining PGAL gets recycled to form more RUBP molecules to continue the cycle. And again, energy is required from ATP for that step. This is just a quick diagram showing the chemicals that are needed in the Calvin-Benson cycle. All right, and lastly, we'll just do this quick question. We are given this diagram and we have some missing parts that we have to fill in. So each letter represents a missing component. We're going to match the missing component on the right with the correct letter on the diagram. All right, so let's start with glucose first. Where in this diagram is glucose being produced? It is here at E. After the PGAL, some of it is used to produce glucose, and some of that will continue on to form more RUBP. The carbon dioxide is here in A, where it first enters the cycle. ATP is here. ADP is here, when 2 of those plus 2 phosphates. And lastly is D, which is right here. How many molecules of PGAL? It is 2. 2 molecules of PGAL will go for the formation of, or the synthesis of glucose, and then 10 of the PGAL molecules go to the formation of more RUBP molecules. All right, so in summary then, the Calvin-Benson cycle is the last step in photosynthesis, and it is necessary because it uses the products from photosystem 1 and 2 to fix carbon. So atmospheric carbon dioxide gas binds with that RUBP molecule to form that 6-carbon molecule, which is very unstable. It'll form the 2 3-carbon PGAL molecules. Then ATP and NADPH provide the energy and reducing power to form PGAL. They will then leave the Calvin-Benson cycle to form glucose. The other molecules will continue to be recycled. ATP is required for the PGAL bonds to be broken and to make RUBP. And this part of photosynthesis is responsible for removing carbon dioxide, which is a greenhouse gas, from the atmosphere. It's also responsible for the creation of biomass. Some applications of light-independent reactions may lead to the colonization of space, reduction of global warming, or even increased crop production.